Okay, so welcome everyone again to the Green Era Hub webinar series. My name is Barry Crosby, and I'm one of the communication officers for the Green Era Hub project. If you've been here with us before, you'll know that the Green Era Hub project officially started in September 2022 as a coordination and support action of the European Commission under the Horizon Europe programme, and it currently represents 15 EU ERA-NETs, co-funds, and self-sustained initiatives in the field of agri-food biotechnology. We started these breakfast clubs here in October last year, and we've had 5 very successful series so far. The first was Circularity in Agriculture. The second series was related to biotechnology to advance sustainable solutions for bioeconomy. The third was Sustainable Food Systems. The fourth looked at precision agriculture technologies for a sustainable future. And the one that we just finished actually this month was plant health and protection. If you're interested in listening to the talks related to any of the topics from these series just mentioned, our recordings are now available to view on our Green Era Hub YouTube channel. I'm very excited to say that today marks the beginning of our new Breakfast Club webinar series in which we're going to be focused on the topic of animal health and welfare. So for the next 3 months or so, we will host a series of expert-led discussions aimed at fostering a deeper understanding of sustainable strategies for improving animal health, preventing disease, managing pests, and strengthening the connection between animal, human, and environmental well-being. Just a few housekeeping announcements. Please feel free to type your questions in the Q&A chat section, which I've enabled for everyone joining us today. I'm going to do my best to address as many questions as possible within the time that we have today, which is 1 hour. So if at the end of the session you have a question for either of our speakers, please do remember to address the speaker in the question. Now, today we have 2 panelists joining us, Professor Christine Van Rieth and Professor Tom McNeely. And we'll open our session this morning with Christine Van Rieth, leading veterinary virologist at the Faculty of Veterinary Medicine within the Department of Translational Physiology, Infectology and Public Health at Ghent University. Specializing in swine influenza viruses and porcine respiratory coronavirus, Professor Van Rieth's work focuses on understanding the pathogenesis and immunobiology of influenza and PRCV in pigs, the role of swine as potential reservoirs for pandemic influenza and coronaviruses, and the development of broadly protective vaccination strategies. Overseeing a team of dedicated researchers at Ghent University, Professor Van Rieth also leads efforts in the surveillance of influenza and coronaviruses in cattle. With over 160 publications and active involvement in multiple interdisciplinary and international collaborations, she's widely recognized as an expert in her field and in her efforts to bridge the worlds of veterinary virology and public health, particularly within the context of the One Health framework. Today, Professor Van Rieth's talk is entitled Emerging Porcine Influenza and Coronaviruses, which is part of the EU-funded International Coordination of Research on Infectious Animal Diseases, ICRAD initiative. Christine, thank you very much for accepting our invitation for a webinar series. I'm going to stop sharing my screen now so you can begin sharing your presentation. Thank you very much.
Very much for this kind introduction, and welcome to all. This project is about influenza and coronaviruses of swine. And these virus families have caused some of the deadliest pandemics in human history. And I'm sure all of you will vividly remember the COVID-19 pandemic. But 10 years earlier, we had an influenza pandemic. And the origin of this pandemic virus is shown here. The genetic material of the virus is inside the virus particle. It consists of 8 separate RNA segments, and we use different colors for gene segments of different origins. So this novel pandemic virus came from swine. It was a reassortant, a sort of hybrid virus, with genetic material from two well-known swine influenza viruses, one European virus and another virus from North America. And unlike other swine influenza viruses, this virus could spread easily between humans. And it also became widespread in swine populations worldwide, and it changed the epidemiology of swine influenza. I must point out that swine are true mixing vessels for influenza viruses. Simultaneous infections with different viruses are occurring all the time, and these viruses then swap genes to create novel reassortant viruses. And on this slide, we have the 2009 pandemic virus in a different color now, and the European avian-like swine influenza virus in green. The other four viruses are novel reassortants between these two parental viruses. And you will notice that they have part of their genetic material from the pandemic virus. And in theory, this could make them better fit to replicate in humans than the original avian-like virus. But this is just a theory. And therefore, we decided to study these 6 swine influenza A viruses together with the swine influenza D virus and a porcine respiratory coronavirus. All these viruses replicate in the respiratory tract of swine, and their transmission is airborne. Influenza D is a bit of an outsider. It's a different influenza genotype. The natural host are cattle. It may transmit less efficiently between swine, and there is discussion as to whether it may transmit from swine to humans. All other viruses are very widespread in swine in Europe. Swine influenza A may be more important than PRCV from the clinical viewpoint, and it's the single virus with proven zoonotic and pandemic potential. So our central hypothesis is that these 8 viruses will differ in each of the aspects shown here: their transmission dynamics, pathogenesis and immune response, and zoonotic potential. And we are studying each of these topics in a separate work package of the project, work package 1, 2, and 3. The comparison between the viruses is very important, and in work package 4, we are applying mathematical models on all the data from the previous work packages, and this should allow us to make predictions about the behavior of existing and emerging viruses. So in this very brief presentation, I will focus mainly on the first work package, which is about transmission. And we know since long that influenza and coronaviruses transmit via respiratory droplets of various sizes in the air, also via contact with infected persons and via virus-contaminated vomits. But there remain so many unknowns. For example, which of these 3 transmission routes is most important? How far can these particles travel? How long can the virus remain infectious in different environments? And the COVID-19 pandemic has very impressively demonstrated our ignorance about transmission. And for a better understanding, we need experimental studies under very strict and standardized conditions, and preferably in the natural virus host. And this is the task of my PhD student, Huan Huo. So she's performing transmission experiments with 24 piglets each time. 8 pigs are inoculated directly with a given virus in the nose, and 2 days later, contact animals are introduced in the same facility. Direct contact pigs are placed in the same pen as these donor pigs. Indirect contact pigs are in the same stable, but in a separate pen. And we also use ferrets. Ferrets are highly susceptible to human and zoonotic influenza viruses. And in influenza research, they are used as a surrogate for humans. We collect many different samples. Nasal swabs are collected daily from each individual animal, and as many as 18 environmental samples are collected every other day. So this is a layout and a top view of the stable. This is the pen of the donor pigs and the direct contact pigs, and the indirect contacts are in a separate pen 6 meters away. So this means that they can only become infected via a virus in the air, and the ferrets are here. The numbers are the locations and the items for the environmental samples. And we have samples in red, and these have direct contacts with the infected pigs. You can see them here. They include feeders, drinkers, also chewing ropes for the pigs, and gloves and boots of the caretakers. But we also sample places out of reach of the animals, and these are the green numbers. And these samples can only become contaminated via the air. And so far, we have done experiments with 3 different viruses, the coronavirus and the 2 most important swine influenza viruses. And the graphs here are virus titers in nasal swabs of the pigs, the amount of infectious virus in the nasal swabs. They are group means, and the upper graphs are the directly inoculated pigs in different colors for the different viruses, and the direct contact pigs. The lower graphs are the indirect contacts. So you will notice that virus shedding in nasal secretions starts as soon as 1 day post-inoculation of the pigs and 2 or 3 days post-contact in the direct contacts. And another few days later, we can detect virus in the indirect contact pigs. So all 3 viruses manage to transmit by contact and by the air. But there are also some subtle differences. For example, shedding is a little more prolonged with the coronavirus. And it is shortest and highest with the avian-like swine influenza virus. And it seems like this virus is spreading best through the air. Another important finding is that only the influenza viruses could transmit from the pigs to the ferrets. And we could detect all 3 viruses in environmental samples, but only in a few samples and a few time points, and mainly in the items in direct contact with the pigs, and the amounts of virus were rather low, around the detection limit. The second work package is supervised by Elma Chillian at the Purbright Institute, and they are inoculating pigs with the 4 most important viruses of the project. And they are looking at what is happening inside the pig in terms of virus replication, pathology, and immune response. So they must euthanize piglets at different times post-infection to collect samples of the respiratory tract, blood for white blood cells, and the spleen. And they are using several very sophisticated immunological assays. Maria Montoya's group in Madrid is going one step further. She is working with individual cells from the infected animals with dendritic cells, and they are trying to obtain information about the abundance of multiple messenger RNA transcripts. And you can imagine that they obtain very large and complex data sets. And these are just some preliminary data from pigs infected with the coronavirus and the 2009 pandemic influenza virus. And the main finding is that the altered pathways differ qualitatively and quantitatively between those two viruses. The third work package is about zoonotic potential. Can these viruses infect humans, and how well? So here we must rely on in vitro models of the human airways and cell culture. And most of the work is done by Xander de Haan's group at Utrecht University. They are specializing in virus receptor interactions. So influenza A viruses use sialic acid receptors to bind to host cells and enter the cells. But these receptors come in different sorts. The detailed structure will differ depending on the animal species and the location in the body or in the respiratory tract. And it's well known that influenza virus from birds prefer to bind to different receptors as compared to the human viruses. And this is one reason why avian influenza viruses rarely infect humans. And in Utrecht, they are studying two important virus properties that depend on the nature of the surface proteins of the virus. The binding to a whole range of sialic acid receptors and the release of the virus from these receptors. So unfortunately, we don't have time to go into details, so I have summarized a few important findings from work package 2 and 3. The porcine respiratory coronavirus seems to cause the most prolonged virus shedding and the highest lung viral load. The most severe lung inflammatory reactions were observed with the avian-like swine influenza A virus. And the swine influenza D virus gave the mildest lung lesions and minimal numbers of cytokine-secreting cells in lung lavage. And this suggests that this virus is really less pathogenic for pigs. And in Utrecht, they found different receptor binding preferences for swine influenza A viruses with different surface proteins. The fourth work package is the mathematical modeling. And here we have one team from University of Leeds in the UK and another team from Comillas University in Madrid. And they have to wait for the data from the other work packages, and they are now gradually starting their work. So in Leeds, they focus on the viral dynamics and the immune response and the comparison between the different viruses. And in Madrid, the emphasis is on the transmission dynamics and the role played by the different transmission routes. And as a virologist, I'm very unfamiliar with the modeling. It's difficult for me, and it's even hard to believe that mathematics can predict the behavior of viruses. But it's exciting It's for sure an added value to the project, and the future will tell. And finally, I want to thank our funder, ICRED, and all the great members of this consortium. This picture has been taken during our first in-person meeting in London in January of this year. And I also thank you for being present, and I will be happy to take any questions.
Christian, thank you very much for such an interesting, insightful presentation. I just want to remind everyone that the chat box and Q&A section is open, so please do feel free to type a question for Professor Van Rees at any point. Christian, just while we're waiting for questions to come in, I had one or two questions of my own, if you don't mind. I'm just fascinated about the transmission process of both the influenza and coronaviruses in swine. And I'm just kind of wondering what factors most influence the transmissibility of swine influenza viruses between pigs? And indeed from pigs to ferrets. You mentioned just at the beginning of the talk.
That's exactly what we are trying to find out in this project. It's a very difficult question, and it's a multifactorial question. It depends, first of all, on the amount of virus that is produced by the individual infected pig. It also depends on the sites of virus replication inside the pig. For a very efficient transmission, the virus must replicate in the upper airways, in the nose, not only in the lungs, but also in the nose. And the portal of virus entry in the naive pig should also be the nose. So transmission in the upper airways is for sure a requirement. Yeah, it also depends on the stability of the virus in the environment, because viruses will rapidly become inactivated outside the host. And for transmission, they have to survive for a little while in the air. And then the receptors they use are important. Yeah, there are so many factors, and that's exactly the topic of the project and what we are going to try to model. And we do believe in the differences between different viruses that will also depend on the exact composition of the virus. Yeah.
Sound very good. And Christine, just in relation to the conditions that pigs are kept in typical farm situations, I was just wondering about um, you know, the issue of housing density or different kind of farm management techniques and how they impact or affect the risk of influenza and coronavirus kind of spillover from pigs. Have you kind of would you be able to kind of say anything about that for our
That's
attendees today?
again, that's a very good question. Housing density is very important because the more pigs on 1 square meter, the higher the density, the more virus there will be in the air, and the more risk for transmission. And this risk is reduced by ventilation. That has also been shown in several other studies. The more air cycles per hour, the more virus that will be cleared from the air, and that sort of reduces the transmission. So, yeah, farmers can sort of influence virus transmission to some extent by keeping fewer pigs in one building and by having a better ventilation. Yeah. But in
Excellent.
our experimental conditions, this is why we have to standardize these factors, always using the same number of pigs in one experiment, the same experimental design, and the same environmental conditions. In our case, it is 8 cycles per hour ventilation and the same room temperature, the same relative humidity, because all these factors can influence the transmission and the survival of infectious virus particles in the air.
Fascinating stuff, Christine. Thank you very much. And just one final question from me, if you don't mind. I was struck very much when you were speaking about the interdisciplinary nature of the study. And I was wondering, it may be too early to tell at this stage because the project is still ongoing. But I was wondering how has collaboration across disciplines, particularly in the context of like virology, epidemiology, and there you mentioned at the end computational modeling, mathematics. How has that shaped, do you think, or improved the outcomes of the research?
Um, it's, I believe it is this multidisciplinary collaboration is something rather recent,
Mm.
and it is one of the positive side effects of the COVID-19 pandemic, I think. And it's for sure an added value, but how important it is, the future will tell us, I believe. Yeah,
Absolutely.
I'm also a little skeptical as to whether everything can be modeled by mathematics, but it's exciting, and our project is perfect um, to prove the added value of the mathematical modeling. Yeah.
Absolutely, and very much breaking new ground, I suspect. Christine, thank you so much. I mean, I could go on and ask so many questions about this fascinating research. Unfortunately, due to time, we've got to move on to our next speaker. But thank you very much for providing us with such a kind of comprehensive and wonderful insight into the project, and the very best wishes to you and to your team. Okay, so now joining us is Professor Tom McNeely, Scientific Director of the Morden Research Institute and Chief Executive of the Morden Foundation in the UK. A qualified veterinary surgeon, he completed his PhD at the Royal School of Veterinary Studies, University of Edinburgh, where he now holds an honorary professorship in the College of Medicine and Veterinary Medicine. Professor McNeely's research focuses on mucosal immunology in ruminants, with a particular interest in host pathogen interactions at mucosal surfaces and how these interactions can guide the development of next generation disease control strategies. His work spans a range of experimental systems, from cell culture and organoids to tissue explants and in vivo challenge models, to investigate immune responses in the context of parasitic and bacterial infections. He leads research on the development of vaccines against key veterinary and zoonotic pathogens, including gastrointestinal parasites in cattle and sheep, which has attracted widespread support from major funding bodies, including the BBSRC, Wellcome Trust, among many others. Today, Professor McNeely's talk is entitled QNet Assess, Development of a Pan-European Network to Understand How Coxiella burnetii Genotype Determines Pathogenicity and Zoonotic Potential. Tom, thank you very much for accepting our invitation to our webinar series. We're absolutely delighted to have you joining us today. So I think without further ado, the floor is yours. I'm going to stop sharing my screen and you can start sharing yours. Thank you very much.
Great, thank you very much. I'll just share my screen and get the pointer. There's a pointer. So hopefully everyone can see that. So the eagle-eyed amongst you will realize that my biography is very much related to immunology and sort of my interest in vaccine development. So you might ask the question, why am I coordinating a project on Coxiella genomics? And this is really because when we're studying vaccines and vaccine development, we want to really have a good understanding of the likely coverage of a vaccine in a population, particularly if we are looking towards development of subunit vaccines. Some of my previous work on E. coli that produce Shiga toxins, so Shiga toxigenic E. coli's, where we can develop, excuse me, very highly effective subunit vaccines. We actually have a plethora of knowledge about the genomics of these S-Tech strains. Whereas in Coxiella, as you'll see as I go through the slides, we have very little understanding so far about the genomics of Coxiella in the field, and there are good reasons for this. I'm just going to talk to you about Q fever, the background of the disease. So it's a very successful pathogen. It's distributed globally and it's been detected in every country except Antarctica and New Zealand. It's an obligate intracellular gram-negative bacteria that causes the disease. We know that cattle, sheep, and goats, and in particularly the small ruminants, and in particular goats, are the main reservoir of Coxiella burnetii infection. And this is the source of infection in humans. The difficulty that we have with Coxiella infections is that it's the case both in humans and in animals that most infections are asymptomatic, but we can get quite spectacular clinical disease. We can get abortion storms, particularly in goats, less so in sheep, and even less so in cattle. But it can cause birth of weak offspring or more subtle clinical signs such as milk drop or rumbling infertility. And there's a lot of controversy about the role of Coxiella in repeat breeding, for example, in cattle and metritis. We have a very high prevalence in our ruminant populations. And this is a very interesting result that we see very high levels of exposure. If we do serosurveys or we look at Coxiella presence within, for example, bulk milk tanks, we can find almost all bulk milk tanks in the UK and across Europe will be positive. But we have very little clinical disease in, for example, in our animals that we can actually confirm a diagnosis and the case in humans. So we might get a few hundred cases in humans across Europe, despite this very high prevalence. However, while most diseases in humans will be sort of a very sort of acute, non-specific febrile illness, which resolves by itself, in a small proportion of cases, they can develop a chronic form of the disease where the bacteria is not cleared, and it often lodges in the heart valves, causing endocarditis and requiring the removal of and replacement of the heart valves. So it can cause quite severe morbidity, long-lived morbidity. And also it can cause quite spectacular human outbreaks. For example, one in the Netherlands between 2007 and 2010. And just to give you a flavor of the types of outbreaks and the sources of infections. So in the UK, in Wales, there was an outbreak in 2002 where over a third of the workers in a cardboard manufacturing plant were affected. And we thought that this was to do with contaminated straw in the building. A probably more direct kind of link here, abattoir workers working with sheep through the abattoir. 110 cases there, of which over half were symptomatic. In England, we had an outbreak of 30 cases, which had a very high hospitalization rate, and that was thought to be due to airborne infection. And in the Netherlands, as I mentioned before, a very big outbreak in the Netherlands, which was linked with dairy goat farms. And that's then instigated a nationwide control program in dairy goats and in dairy sheep, in which there is compulsory vaccination. So it can cause quite spectacular human disease outbreaks. Yet I've mentioned before that a lot of the infections that we detect were either missed or are asymptomatic, so very mild. So I think I've hopefully convinced you that it's an important livestock and human pathogen. But we really have very limited understanding of the genetic determinants of the host specificity and virulence. And this means that it's very hard if we detect Coxiella in the environment or on a farm to assess the likelihood of a spillover into humans. And this is largely due to the fact, as I mentioned before, we have very little genomic data from different hosts and associated good clinical metadata. And this is because diagnosis is mainly based on histopathology, smears, serology, and molecular methods such as PCR, but we don't routinely isolate because it is very difficult to isolate this bacteria from clinical samples. So essentially, we have very limited information in terms of complete whole genome sequences to start doing some genome association studies to try and get a better handle on the genomics. Just to sort of say, we do know something about this from sort of from some limited whole genome sequence data. So it seems to be there's 6 genomic groups. There's a core genome of about just over 1,000 genes, and it carries 4 plasmids, or a range of different plasmids, of which we've detected 4 so far. One thing that's very interesting about this is that the zoonotic potential does seem to differ between the source of Coxiella from different ruminant species. So, for example, if we look in this graph here, cattle are in blue, and human infections, human strains isolated from humans are in yellow. And you can see that in the cattle, there's very little overlap or relationship between human strains and cattle strains, whereas most of the human infections kind of lie within sort of goat or sheep strains. So we think that maybe cattle strains are going to be less pathogenic to humans and have less zoonotic potential. Also, we're very interested in chromosomal rearrangements. This is the 9-mile reference genome. And the Coxiella genome is quite small. It's just over 2,000 genes, as I mentioned before, usually. It has lots of repeated sequences, which means that the genome can flip in terms of with chromosomal rearrangements. So you can get sort of a big reassortments of the genome. This is the Netherlands outbreak strain, which has lots of genome reassortments. And this is a cattle strain, which is not thought to be pathogenic. So we think that something is happening in these rearrangements of the chromosome, which increases pathogenic potential. So this is the project. We wanted to generate a really comprehensive understanding of genetic variation of Coxiella at the whole genome sequence level, and then how we can use this information to assess zoonotic risk, livestock pathogenicity, and inform Q fever control, for example, making certain any developed subunit vaccines are going to have broad coverage of different Coxiella strains. This is a consortium. We wanted to try and generate a consortium that involved all the Q fever reference laboratories, or as many as possible within Europe. And we also have hop-on partners that are joining the consortium. We have actually even more hop-on partners in this because we've just gathered a lot of the human reference laboratories as well. This is important because if you think the number of confirmed cases per year is in the tens to less than 100 per member state, for us to get a good biobank for isolation or for sequencing, then we need to have a very wide consortium. This is the project. So we have work package 1, which is all about collating the biobanks and ensuring appropriate metadata. I mentioned before, it's very difficult to isolate Coxiella, so we have a work package looking at better methods of isolating Coxiella from clinical samples, but also looking at direct sequencing of Coxiella from clinical samples. We've got the sort of the engine room, which is basically the genomic sequencing, and then we're going to do functional genomics and genome association studies in work package 4. So this is in 2 sort of streams. One is looking at genetic determinants of virulence and host range, but also can we have some phenotypic assays that are simple to conduct in the laboratory to give us an idea of virulence of that particular strain. And then the idea is to bring it all together to sort of to establish a legacy of the project, a sort of a pan-European monitoring and genotyping network. So I'm briefly going to talk just a few highlights about the work packages. So we've actually been very successful in collating a big biobank of clinical samples, and we've determined the minimum associated metadata that is required for each sample. This is going to be shared, as I mentioned before. This is the footprint for the ongoing genomic surveillance. But we've also made certain that we've tried to harmonize guidelines for diagnosis of coccidiosis, the disease in ruminants across Europe. And this is a paper that's just been published in Open Research Europe. A really holy grail is really to try and get the genomic information from a clinical sample. It's very hard to isolate these bacteria from clinical samples, often because they're just at very low bacterial loads. But what we can do is use direct sequencing of clinical samples. And this is work done by Rohe Day. And basically what they've done is they've developed a selective whole genome amplification method in which you basically selectively amplify across the whole of the Coxiella genome to allow you to get good coverage of the Coxiella genome, even in a sample with relatively low levels of bacteria. And there's various different methods of using the MinION approach here, but we've got a sort of a protocol now that has been shared across the different member states, and we can actually get closed genomes directly from the clinical sample, which I think actually probably is a game changer in terms of this project. This is just an example of a successful whole genome sequence that's been retrieved from a clinical sample from 19 contigs. It pulls out chromosomal and plasmids, and we've determined that the limit for samples to go through this pipeline is a CT, that's a sort of a measure of abundance based on a quantitative PCR of less than 25 cycles. The other thing that's really interesting about this direct MinION sequencing is you can pull out the Coxiella genomes, which are in blue, but you can also pull out co-infecting pathogens as well, for example, E. coli and chlamydia. And I think this is also going to make quite a game changer in terms of our understanding of abortions in ruminants, particularly because a lot of our abortions retrieve no confirmed diagnosis. Optimizing isolation protocols. This is the standard way of doing it, in vivo isolation of mice, and then you amplify in chicken eggs. But we want to move to a system which is much more efficient, uses less animals, and it's where you take the sample, which will be highly contaminated with other bacteria, homogenize it, somehow filter it to reduce the contamination, and cultivate either in cell culture or xenic media. And then this will allow us then to sort of perform whole genome sequences. So we want to identify samples to limit the contamination, enrich Coxiella, and facilitate cultivation. And one approach is to try and improve the xenic media. So, for example, we know that certain genomic groups are very poor at growing xenic media. And this is just to show some work that's been done by Laura Flute, a PhD student at Sansano, where she's looking at different media formulations and tried all these additional additives to try and improve the growth of Coxiella within these media formulations using 3 different genomic groups. And you can see here that ACCMD, which is one of the proprietary media, and ACM2 differ in their ability to grow these 2 different genogroups, but not this one here. We haven't found any particular additives that have improved the growth of hard-to-grow Coxiella, but we're doing metabolic pathway analysis from the whole genome sequencing data that we've generated and doing high-throughput phenotypic microarrays, which allow us to test additional metabolites in a very high-throughput manner. This is just a very brief summary from Selina Fuchs's PhD to show that she's done a huge amount of work on the pre-treatment of the samples, including heat treatment, antibiotic treatment to clean them up, enzymatic treatment. We found to be very unsuccessful, and they lost a lot of bacteria using enzymatic treatment, but better filtration and homogenization steps, the best type of cells to use, which are viewer cells, and the antibiotics post-treatment to make certain we only grow Coxiella. And this has been very successful and it's been rolled out to the group. The genomic sequencing, we had to go back to basics here. We use Microbes NG, which is a facility in the UK for all our sequencing. But we had to validate a lysis buffer to allow our partners to prepare Coxiella strains, put them into lysis buffer and ship them to Microbes NG. It's a hazard group 3 pathogen, so we needed to make sure that the lysis buffer that is provided by Microbes NG kills the Coxiella, which indeed it does. And this is where we're at. We've sequenced 100 strains in year 1, and you can see the split between the different countries here and the different types of origin of the strain. We also have 100 strains that we're going to be sequenced in year 2. And this is just to give you an update of where we are here in terms of these are all the samples that we've collected. Below this threshold here is where we're going to be isolating from. And this is the CT value of 25 in which we can do direct sequencing. So we're going to be pushing a lot of strains through the direct sequencing. And hopefully we'll get even more than the 200 genomes that we were predicting to get at the end of the project. This is just a shot of the functional genomics of the Coxiella burnetii isolates. And it's showing that we pull out 9 different clusters. This is the latest phylogeny. So this is the most up-to-date phylogeny we have of Coxiella to date globally. And what you can see here is we have the ruminant clusters. We have 4 clades. One is cattle and 3 are small ruminants. And you can see that this is the host, which is down here, and the location. So most are from Europe, some are from North America. And just to zoom in on the cattle cluster, what we're seeing here again, if you sort of remember back to when I was talking about the host, so and the cattle and human question, these are the cattle in yellow now, and the humans are blue. We can see again that that there's a poor association between cattle strains and human strains, suggesting that we are seeing the same pattern, that cattle strains potentially are less pathogenic to humans. Functional genomics, I'm not going to talk about this in any great detail, just to say that there's lots of work ongoing with a PhD student at the FLI. And what they're doing is they're looking at phenotyping Coxiella burnetii in terms of their replication fitness, invasion efficiency, and immune response, using a different range of sort of primary and cell lines from ruminants. And then they also wanted to, this is quite low throughput, and they also are setting up a whole blood infection model to assess Coxiella host infection in a more rapid way. And this would be a sort of looking at sort of expression of cytokines and bacterial killing. And it was just very interesting to see that when you infect whole blood with Coxiella, it's all the leukocytes that are infected, and this is looking promising as a sort of more rapid phenotypic assay. Finally, my last slide is just about how we're going to go about establishing this pan-European network. I think one of the benefits of this consortium and the funding that's allowed us to do this is to bring in all the reference laboratories within at least Western Europe and the UK. And this gives us quite a lot of power to propose recommendations in terms of surveillance. We are have a workflow for surveillance sampling that is going to be taken forwards beyond the end of this project. So how a lab should be biobanking samples, standardized SOPs and workflows. And then where's all this data going to go? Well, we're going to be using the ENA Archives Pathogens Collection portal. This is a sort of a very well-funded part of EMBL. So all the genomic sequence, all the metadata will be uploaded onto this portal for future research activities. So that's a real whiz talk through Coxiella Genomics and the project. Happy to answer any questions, and just to acknowledge a lot of all the groups that are involved here, all the people that did all the work, and our funders, our national funders, and thank you very much. This is a link to the project website if you want any more information, and I'll stop there. Thank you.
Thank you very much. Such an interesting and insightful presentation. I just want to remind everyone that the chat box and Q&A section is open, so please do feel free to type a question for Professor McNeely at any point. Tom, if I could just start off the questions, if I may. I was just struck by something you said at the very beginning of your talk about how there were 2 areas globally where Coxiella has not been identified. One was Antarctica and the other was New Zealand. And Antarctica is fairly self-explanatory, but New Zealand, what are the conditions in New Zealand for Coxiella not to have been identified there?
It's a very bizarre observation. So I guess either it's real or it's never been detected and it possibly is there. So the thing that's very interesting about the New Zealand result is that Coxiella was first isolated in abattoir workers in Australia. So that was in the 1930s. That's where it was first identified as an issue. And the Q is basically because they were seeing abattoir workers with fevers and they didn't know what was causing it, so they called it query fever, and then that shortened to Q fever. So Coxiella is very widespread in Australia, to the point that Australia is the only country that has a licensed human vaccine. But the vaccine is tricky to use and it causes severe injection site reactions in people that have been sensitized to Coxiella. So if you've been exposed to Coxiella, you will not be allowed to get the vaccine. So they use it for vets and abattoir workers, high risk individuals in Australia providing you have a negative blood test for antibodies and you're negative on a skin test for your cellular immune response, a bit like TB testing. So it is rife in Australia and it's rife in the animal population. So I don't understand why it's not in New Zealand. These Coxiellas are very similar. These are kind of almost like symbionts that are present in lots of different species. That's why it has such a host range. So it's just this is adapted to infect humans and ruminants causing clinical disease. I don't have a great answer for you. I don't think anybody really knows.
Yeah, fascinating stuff. Worthy, I think, of a of investigation itself
Yeah.
by some aspiring PhD student. Tom, just a quick question coming in there from one of our attendees, from Matteo. He says, "Excellent presentation. I did not catch if cow isolates have never been associated with human cases."
So I think there are some human there are some strains that have been isolated from human cases which look genetically similar to cattle isolates, but that's generally a rare occurrence. You would predict that the ruminant strains would be sort of transmissible between the ruminant species. But that doesn't really seem to be the case. It seems to be that the cattle have a different sort of subset of strains that are circulating within their populations. We really don't understand the transmission dynamics between sheep, cattle, and goats. But I guess the observation is that regardless of the study, we always see a very strong association in terms of the relationship and the similarity between strains that are identified in sheep and goats and in the strains that we isolate from human clinical cases, whereas we don't really see that with cattle. And this is one of the drivers of this project, is that we don't really understand why that is the case. There's been some more recent work suggesting that some of the plasmids, there are 4 plasmids that have been identified in Coxiella, and there's an association between human disease severity and these plasmids. Now, whether there's some, whether it's the chromosomal genome or whether it's the plasmids that are different between the cattle strains and the goat strains, and that determines somehow the zoonotic potential, we don't really know. So I guess that's what we're trying to understand. And I think once we develop better tools to genotype these bacteria, then we can start looking at sort of holdings with sheep and cattle and goats and see how, do the same strains transmit between the different species, in which case then if that is the case, then it makes it even more difficult to understand why humans are human strains humans don't seem to be infected by cattle. But the exposure rate would be quite high, I think, for people with cattle and goats and sheep, because we know that cattle can shed very high levels of Coxiella just postpartum. Within the first week postpartum, they shed very high levels, even though they can be asymptomatic. So there must be exposure. So I don't think the lack of a link between cattle and human is due to exposure. It seems to be somehow genetically determined by the bacteria, but we don't know why.
Thanks, Tom. Katie is just asking if the slides can be shared. I'm not sure, Tom, if there's a facility to do that from your end, but I would say to Katie that these webinars are recorded and are, with the consent, obviously, of the panelists, are uploaded to the Green Era Hub YouTube channel. So you'll be able to review those at a later stage, Katie. But sorry, Tom, come in there if you have anything to add to that.
No, that would be fine. Yeah. So I can basically PDF the slide and send it to you.
Perfect. Thank you so much. And Tom, just very lastly, can I just ask you a little bit about the collaboration and kind of network integration idea, which I think is a really important component of this research project. I was struck during your presentation by the scale of the consortium, and I'm very interested in the development of this kind of pan-European Coxiella surveillance network. I was just wondering, for the benefit of our PhD students joining us, particularly in postdocs. If you could maybe just talk a little bit about the biggest benefits, maybe even some of the obstacles that you have and your team has encountered in the creation or attempted creation of a pan-European Coxiella surveillance network.
Yeah, so I would say that the reason that we have such a large network is, I guess, is twofold. One is just very practical reasons, is that we kind of know how many confirmed clinical cases we get per year from each of these member state countries. So, for example, if we were to try to do this just in UK or just two countries, we would not have enough. We would not have enough samples that would be have potential for isolation. So that was so it was basically we needed sort of this scale to compared to, for example, doing genomic surveillance of E. coli, for example, or STEC, very easy to isolate. You can isolate them from sort of lots of different samples, and you could have thousands of genomes if you wanted to quite rapidly. Whereas for Coxiella, because it's very hard to isolate the low, confirmed clinical cases and so many asymptomatic cases that will be missed, is that we needed that scale of consortium to really address some of these questions, because the previous studies were all, fairly small scale. So they weren't sufficiently powered for us to start, addressing some of these questions around the link between the genomics and the sort of virulence and host specificity. So as a practical reason for that, it is harder to manage. It's it's a large consortium, and with the nature of it, because I think there's been a will, or it's been a very well known knowledge gap in Coxiella, is that we just have so little information about the genomics diversity of Coxiella, that now we're getting a lot of requests for hop-on partners. So we probably have another 6 or 7 partners that have hopped onto the project since it started. And I think that shows that there was we're filling a sort of currently an unmet need. It does make the coordination sometimes quite tricky, because it's a very large consortium. But I think actually being at this scale means that we've captured all the reference laboratories and what I want to do is make certain that this is almost the start of the Coxiella genomics surveillance approach, is that we embed these protocols within reference laboratories. So beyond this project, Coxiella, genomic, isolation and sequencing and genotyping of Coxiella becomes standardized across Europe, and the more strange you have, the more genetic information with the right metadata. So also making certain we get standardized metadata. That means that we will probably be more and more able to answer some of these key questions around Coxiella and its genetic diversity. So I think it was partly because we knew that we needed a large consortium to get a number of samples to do some meaningful work. But also it was, the reason that a lot of people wanted to join the consortium was because they realized this was an important sort of area to get into. And it was also very timely, because Q fever had just become, when we were starting to commission this project, Coxiella was just becoming a reportable disease in the EU and in UK. So that any Q fever, cases in ruminants and in humans had to be reported. So it was very timely.
Absolutely. Tom, thank you very much for, as I say, for joining us and for giving us such an excellent overview of the project. And we wish you the very best, you and your team, the very best with it over the coming months. I'd like to thank once again our excellent panelists this morning, Professor Christine Van Rieth and Professor Tom McNeely, for giving us these wonderful and insightful presentations and providing us, as I say, with wonderful overviews of the respective projects this morning. Unfortunately, we've just run out of time, so we'll have to leave it at that. But our next webinar will be on May the 8th. I'll be sending out the invitations to all of you in the coming days. In that breakfast club, we will have Professor Jane Hodgkinson, Professor of Molecular Veterinary Parasitology from the University of Liverpool, talking about mapping resistance loci and interrogating mechanisms of tricholabendazole resistance in European isolates of Fasciola hepatica, and Professor Peter Geldof from the Faculty of Veterinary Medicine at Ghent University, discussing plant-based production of anti-nematode vaccines. Again, thank you very much to our panelists for joining us this morning, and thank you for everybody for attending and for your questions. We really hope you enjoyed the webinar, and we hope to see you again with us next time. Thank you very much, everybody, and have a good morning.
Thanks, everyone. Enjoyed that. Thank you.
Thank you. Bye-bye.
Thank you. Bye-bye.